again, we're going to be hearing today from Ratul about one of the two seed funding awards for the research and tech transfer area. Ratul's part of Iowa State CBE department, Chemical and Biological Engineering, is a relatively new hire, recently joined our department. Seems like recent, it was probably several years ago. And he's part of PDT, the Proteins for Diagnostics and Therapeutics focal area. And he's going to be telling us about his work. And he's presenting early because he does have class today, and he's already moved his schedule around to accommodate this presentation. So thank you, Ratul. Please go ahead.
Yep. Thanks, Laura. Thanks for the intro, and also appreciate the opportunity to present and also opportunity to get the seed grant. So okay, so I'll be talking about like what I proposed and what I plan to do. This has been sort of on the frontier of what we wanted to get towards. So we're trying to understand if we can build a foundational AI model. A foundational AI model means that a model for a situation for which a model does not exist as of yet, right? So to understand how atomic vibrations helps us, by tracking those, we can ultimately explain what's going on inside a full living cell. So obviously the focus is on understanding and designing better enzymes. But okay, before I jump into the talk, I have a shout out to give to the other award winner like Hyungmin Seo, because me and Hyungmin sort of go back a long way. So 7 years back, I think we've known each other through the academic career. He's slightly junior to me, and like we've always, been good friends. So he invited me to University of Iowa last week, and I had a great time there. And strong recommendation for Utopia. Please go and have the yogurt there. That's pretty cool. All right, cool. Let's get back to the stuff. So, okay. So before we get, so the holistic idea behind my lab's work is to understand the inner workings of a living cell, mostly along that metabolic front. So we started targeting understanding enzymes one at a time, and we looked at the large class of enzymes called hydrolases, for which there is the most amount of crystallized structures that are out there. So this is a work in collaboration with Nigel. So where we basically looked at 19,000 hydrolases that have been reported in PDB so far. And we try to inquire as to if we were to figure out how to understand the function of a certain enzyme, how much of the structure do we need to actually study? Because if you study the whole structure, probably somewhere here you can see this whole structure in red, that's not fully responsible for the activity. And more of the structure that you include, your information is going to get more and more dilute. And you're going to find very dissimilarly looking enzymes, which ultimately show very similar functions. And you will not be able to explain what is the core set of similarities between these similarly functioning enzymes. So we created what is called a definition for a reactive core. This was the first part. So where we define these yellow regions over here, where the catalytic activity is happening, and also some of the surrounding amino acids that actually hold that catalytic site in its current shape and form. So we created a database of such subparts of proteins and used that as a metric to quantify similarity of function. And we also put this database and integrated it with the PDB. If you go to Protein Data Bank now, there is a specific way to access structure classification tools. You'll find RC hydrolase over there. And what does this tool allow you to do? It allows you to not only look at proteins that are hydrolases and understand which parts of them are important for the function, but also lets you cluster like similarly looking parts by a certain percentage similarity that you care about. And it will let you download the data and use it for any purpose. One of the key aspects we care about here is what organism is this enzyme getting expressed in. So, if you're trying to engineer a strain, you care about the specific organism. So this information is pretty useful. And the overall goal is that while we understand enzyme structures and we know how to, tweak them, we need to predict how functional they are. Like the prediction of functionality is defined largely by Km and Kcat of a given enzyme. And so we wanted to come up with a way to unify all the pieces of information that are present in sequence and structural databases to create this unified database, which has sequence structure as well as reactive center information, as well as catalytic activity prediction. So we spent some time in the lab over the last 9 months, as this bioarchive article, is indicative of, is we try to set up a machine learning platform to predict the Km and Kcat values of enzymes. So there are a couple of other tools that are out there which do it. The accuracy is not great in the current state of the art. The primary reason for that is Kcat values in enzymes range from 10 to the power -8 to 10 to the power +8. That's 16 orders of magnitude. And the number of data points that you have for which you know experimentally the Kcat values, that's roughly 25,000, 26,000. So that's 10 to the power 4. So if you have 10 to the power 4 data points spread across 10 to the power 16 orders of magnitude, you cannot do regression. So that's what the other tools have been doing, where they wanted to exactly accurately predict what is the Kcat of a certain enzyme. And we sort of go beyond the convention over here and say that, first of all, who cares about the exact value? If you're in an industry, if you're working with an industrial enzyme, you don't really care whether it's a 4.1 multiplied by 10 to the power 4. You just care about whether it's 10 to the power 4 or 10 to the power 5 or 10 to the power 6. So we projected this as a classification problem and said that here are these defined buckets where you have orders of magnitude. As long as we can predict the right order of magnitude of the kcat value, this should be reasonable. And you can see the accuracy values are pretty reasonable. And we obviously explored different types of machine learning architectures here. You can read this article that's up there right now. But today's talk is more about, okay, what happens when we start understanding enzyme activities? You start looking at, like, zoom out a little bit and you look at a pathway. So once you look at a pathway is a cascade of enzymatic reactions. Each reaction is composed of a substrate that the enzyme breaks or joins. So we just predicted using the previous tool what the activity of each enzyme is. So I should be able to ideally put that information at every reaction and figure out how much flux should go through this. But that setup is slightly cumbersome, and we have been looking at, the seed grant looks at a different way of thinking about it. So what is this? So we have, how do people classically do it? So we have enzyme sequence. So we have that sort of gets converted into a set of numbers. Enzyme sequence needs to get to a convert to a set of numbers, and the substrate information also needs to get converted to a set of numbers. You join these two set of numbers, and then that we call as a reaction because it has the enzyme information and the substrate information. And in the previous study, we have trained against the kcat and km values for this reaction encoding. But here what we are proposing is that if we actually have a reaction encoding which has information about the substrate and the enzyme, can we think about making a large language model for a pathway? What does it actually mean? So first of all, language models mean things which can be arranged sequentially. So if you have sequential entities, you can use a language model to train it. For example, a protein sequence has sequential amino acids, so you can train a language model on it. Believe that these are sentences in the language that you speak, right? And so these are individual words that make up these sentences, and every protein sequence is a sentence in that book. Okay, so similarly, you have small molecules. Small molecules are slightly tricky because they form cycles. But if you remember, smile strings make them linear. So once there is sequential linear information, you can sort of convert them into a mathematical object through a language model. So if you could understand what I'm trying to draw at here, the idea is to be able to convert a pathway into a language. So if the whole pathway is a sentence, what is my smallest token? What is the smallest piece of information that will represent a word in that sentence? The smallest unit of information that will represent a word in that sentence is a reaction. So if you string multiple sets of reactions, you'll get a pathway, right? And so first thing for such an objective is that we need to be able to represent every word in this sentence using a fixed number of numbers. Essentially, either it's a 3-dimensional vector or a 4-dimensional vector or a 5, no matter what that number is. For every reaction, you need to be able to convert that to the same fixed dimensions. Otherwise, the learning process does not work. So we need a fixed dimension of enzymes and substrates, which we have shown in the previous case as to how to do it. But we haven't done so far is to capture the structure. We were purely reasoning on sequences, and we were operating on the small molecules. But how do we incorporate information about the structure of an enzyme here? So here in this proposal, what we showed is that there are several ways to look at structure. With the presence of AlphaFold and also PDB known crystal structures, we can capture vibrational modes of information flow within a structure. Let's say if this were an enzyme and the edges represent hydrogen bonds, salt bridges, etc. So the moment one amino acid vibrates, the other one neighboring with it vibrates. This is what is called the allosteric pathway of interaction. And so we basically extract this information. This is already present when we perform molecular dynamic simulations. It's just that we never use it out of the MD simulation. So we are now using that and also converting the structure of an enzyme into a set of fixed alphabets, like, as I said, whether it's 10-dimensional or 15-dimensional and so on and so forth. So we are doing that by saying that, okay, the activity center is here. This is our reaction core work that I just showed in the first slide. So this is the reaction core where all the activity is happening. And you create radial shells around it. So and figure out how many of those amino acids that are part of the enzyme that are falling within each of those shells. You not only take the identity of these amino acids, but you also take information about who are they connected with, what kind of vibrational information they share with their neighbors. And thus you can create sort of, in an abstract manner, a set of numbers that represent an enzyme structure centered around the catalytic site where the activity is happening. So we have imposed this preliminary concept and looked at different types of enzymes, and this helps us, map all these highways of information flow. This is vibrational information flow through an enzyme. Okay, so next element of this is this is that we are creating a multiple specialized expert network. So this is pretty much not a biological or biomolecular network, but it's a question answer network. So essentially, what we would do is that we'd put some questions about structural stability, like calculate binding free energies of various variants and sort of store it in a database in a question answer format. The question answer format would go like this: that okay, what is the binding score with this substrate with the specific variant, and we'll save as an answer the value of this. This question answer format is very important because this is sort of called a question bank, and this is something that could be leveraged by a very recent development in machine learning. Two models that have come up which can handle question answer is DeepSeek as well as Grok v3. So these models actually have shown that once you're doing your training in a model and you have a bank of question and answers which you're also learning from, you can actually query back the model and ask it, okay, I think mutation X is going to be something that I will do on my protein. Can you tell me what it will do? So the model will actually think and sort of look at the question answers that have been already fed to it, and it'll give back saying that, okay, this will increase or decrease activity because it disrupts this hydrogen bonded network. Remember, these information is already pre-fed for other enzymes which it has trained on. It learns from the enzyme similarity for this specific enzyme that we are trying to engineer and tries to map it and give an analogous answer. So, and it also gives more information, like this is because shell 3, like the, far away from the active site is critical because it connects the catalytic residues to the surface where the solvent is, etc. So if the answer is actually correct, your biological intuition, so we have to be a real expert out here. We have to have a biologist at the very core. So we ask questions that you can also yourself answer. So if you think that that is a correct answer, you sort of put in some reward, and that reward could be one. And now, if this answer is wrong, then it's thinking the wrong way. You sort of fix its chain of thought, saying that, nope, that was a wrong way of saying it. The right way of saying it is this. So you actually don't sit down in front of the computer and do this rewarding. You also have a separate database of questions to query it on and have like a for loop go through it to do its reinforcement for the cases that you want to perform. So that's pretty much the proposal. We are going to build this out. We have installed DeepSeek right now in the Iowa State cluster, and we are playing around with it. So we'll see how this progresses and how we can train this and what kind of results come out of this. And hopefully, we still are able to adhere to the same idea that once we have this reaction encoding, which is going to act as a token, like the smallest unit of information for a pathway, we can string multiple of them next to each other to form like a pathway, right? To form a word or form a sentence. So the good thing about this is that for metabolic pathways, every reaction is mass and charge balanced. So we should be able to impose this criteria so that making sure that it's not predicting something random. It should not predict that the glucose plus ATP is giving me a fructose 1,6-diphosphate. It does not adhere to mass balance. It has to go through these intermediate reactions and an extra ATP. So we can do this mass and charge balance checks. We can do the moiety balance check. For example, if there's a phosphate that's getting added, it is coming from some other place which has a phosphate. And also the sum of all pathways is actually the whole cell. This is glycolysis. You have TCA, you have pentose phosphate. You can have all the pathways, and the sum of all the pathways is essentially your whole cell, which is altogether mass and charge and moiety balanced. We hope that this effort will lead to capabilities such that we can currently do number one using things like OptForce, where you predict predict like which reactions you need to upregulate or downregulate to engineer a specific strain, right? But it does not tell you currently, the point one does not tell you currently as to how do you achieve that point, that upregulation or a downregulation in a specific pathway. But if we have specific enzyme sequence level information, structure level information, and biochemistry embedded in it, we should be able to not only give the first step, we shouldn't be able to tell that while you're engineering for a task, you need to upregulate this gene 1 and gene 2, and here is how you do that. You make this specific point mutation to get to that specific target upregulation or downregulation limit that you're looking for. So one last slide I have after this, which basically says that this is still looking at an enzyme directly from the enzyme sequence level. So far, whatever I've showed you just looks at the enzyme sequence and also the enzyme structure and the substrates structure. whenever these engineering outcomes are predicted, it does not still take into account whether a promoter X sequence is actually compatible with the sequence that you predict, whether it will actually express before it is actually functional. So for that, this is outside of our tool set. So this is something somebody else has developed. I discussed this a little bit back in one presentation. So Evo2 is a genome-based chat GPT, you can say. It's been trained on 128,000 genomes. The simple objective, the way we are going to use this is that if you tell us that, okay, this is the bacterial host that I am going to engineer, we can generate a bunch of de novo genomes for this, which shows variability, yet adherence to the same clade. We can figure out which promoter sequence is for your target gene. Let's say in pink, your target gene is in pink. By comparing the genomes and comparing the blue promoter regions, we should be able to tell which promoter sequences tolerate which kind of sequences for expression. So that's where we are at. I just wanted to give a brief overview. Thank you for listening. That's the last slide I have.
Excellent. Thank you for that presentation. Do we have questions for Ratul?
So Ratul, pH and temperature are very critical in terms of metabolism and enzyme kinetics and thermodynamics and that type of thing. Do those parameters have an impact on your analysis, or is there an input for those?
There is. So the KCAT and the KM predictor leverages as much as pH temperature values that are reported in current experimental databases. So temperature is still something that we can track in the sense that we can perform a molecular simulation, like a temperature ramped simulation, to predict how the molecule flexes in and out of proportion for catalysis. But pH is, as you know, it's much harder as a problem to figure out how it cascades through the protein, like the extracellular, sorry, the pH in the surrounding media, how it impacts the surface exposed residues and how the surface exposed residues interact with the neighboring ones. Hopefully, our hope is that if you're able to track the change of partial charges that are induced by the pH, right, and figure out what are the surface exposed residues that are most affected, like histidine, etc., then we can use this pathway type analysis that I said to probably get to the bottom of the catalytic core and say that probably there is a positive impact versus a negative impact here. But I think it'll just be like a 0 and 1 at the moment, nothing super quantitative.
Okay, thanks.
I like this study because identifying, for instance, mutations responsible for enzyme activity is one of the few examples of explainable machine learning. I'm just wondering, I didn't really understand all the details, but I get the feeling that you're trying to create this connected network consisting of multiple other subnetworks, so it will be a fairly large structure. Do you think you have enough training data for this? And Ratul, how are you planning to test this? What would be your benchmarking protocol to kind of assess the accuracy of this approach?
So the first benchmarking approach for predicting whether our model understands enzyme activity at all or not is the first test is it's a hard zero-based test where we take an enzyme and just identify its catalytic residues and just mutate them to alanines to see if the model can predict the catalysis rate should be 0. The KCAT value should be a 0. If the model cannot do it, then there is no reason why we should trust the model. But that's the first sanity check we always do to abolish catalysis and see whether the model still predicts whether there is no catalysis, because mostly models rely on sequence similarity, in which case if you change the catalytic residue, the sequence similarity is still 99% or like very close to similar. But we build in specifically aspects of catalytic importance of individual amino acids. And the second benchmark is, so there is an industrial company, so it's a part of DuPont, if I'm not wrong. So Gen and Core, they actually have released an industrial data set of 1,300 mutations on the same enzyme, on the same substrate, using the same experimental benchtop setup. And they have released the KCAT values and the KM values of this point mutations done at every location on the enzyme up to the catalytic core. And that's typically something we don't include in our training set. The objective is to see if the model can perform reasonably well on that industrial enzyme.
How do you think this will compare to, say, classical transformer model where you you train it and then you fine tune it for this task of identifying catalytic residues?
Yep. So the bioarchive article, which is called Real KCAT, REALKCAT. So the Real KCAT article actually does this whole exercise of looking at an existing there are three current models which are pretty reasonable, like DLKCAT and a few others. We retrain all of these models with our data set and show that, just retraining the model with the new data is not enough. You have to have the architecture built in to understand where, like, this is like a singularity, right? You touch a specific point on an enzyme and the whole enzyme ceases to be an enzyme anymore. So you have to build in those information gradients pretty mindfully. And the article talks about how we do that. So, happy to chat about that in detail.
Hear you. Thank you.
All right, great discussion. Okay, so again, we're going to be hearing today from Ratul about one of the two seed funding awards for the research and tech transfer area. Ratul is part of Iowa State CBE department, chemical and biological engineering. Is a relatively new hire, recently joined our department. Seemed like recent, it was probably several years ago. And he's part of PDT, the Proteins for Diagnostics and Therapeutics focal area. And he's going to be telling us about his work. And he's presenting early because he does have class today, and he's already moved his schedule around to accommodate this presentation. So thank you, Ratul. Please go ahead.
Yep. Thanks, Laura. Thanks for the intro, and also appreciate the opportunity to present and also opportunity to get the seed grant. So okay, so I'll be talking about like what I proposed and what I plan to do. This has been sort of on the frontier of what we wanted to get towards. So we're trying to understand if we can build a foundational AI model. A foundational AI model means that a model for a situation for which a model does not exist as of yet, right? So to understand how atomic vibrations helps us, by tracking those, we can ultimately explain what's going on inside a full living cell. So obviously the focus is on understanding and designing better enzymes. But okay, before I jump into the talk, I have a shout out to give to the other award winner, like Hyungmin Seo, because me and Hyungmin sort of go back a long way. So 7 years back, I think we've known each other through the academic career. He's slightly junior to me, and like we've always, been good friends. So he invited me to University of Iowa last week, and I had a great time there. And strong recommendation for Utopia. Please go and have the yogurt there. That's pretty cool. All right, cool. Let's get back to the stuff. So, okay. So before we get, the holistic idea behind my lab's work is to understand the inner workings of a living cell, mostly along that metabolic front. So we started targeting understanding enzymes one at a time, and we looked at the large class of enzymes called hydrolases, for which there is the most amount of crystallized structures that are out there. So this is a work in collaboration with Nigel. So where we basically looked at